All right, in this video, we're going to continue our discussion of the regulation of transcription in eukaryotes, specifically multicellular organisms. And where we left off was the question here, which is if we look at two different cell types in a multicellular organism, so liver cells and lens cells, we see that they have the exact same genes, right? All cells have all genes, the albumin gene, crystalline gene, albumin gene, crystalline gene. But not all cells express all genes. The lens cells don't need albumin, so they don't express the albumin gene, but they do express the crystalline gene. And the liver cell doesn't need the crystalline gene, so it doesn't express it, but it expresses the albumin gene. In addition, what we notice is that all of the regulatory sequences of all of the genes are identical. The albumin gene here in the lens cell has this red, gray, yellow, and in liver cells also this red, gray, yellow. So then what is it that determines which gene is going to be turned on in a cell? And the answer is the activators that the cells make. Different cell types produce different activators, which means a different set of genes are going to be turned on. The lens cell produces the pink, the gray, and the brown activators, and the liver cell produces the orange, the red, and the gray activators. And so the question is, if all cells of a multicellular organism stem from that original single-celled embryo, the zygote, then where do these differences begin to emerge? Where does asymmetry start? And there's two answers for that. The first is something called cytoplasmic determinants, and the second is called inductive signals. Cytoplasmic determinants. If we look at an egg cell, right? Remember a zygote, here's our zygote, comes about from a sperm fertilizing an egg. Now a sperm doesn't really have any cytoplasm. It's the egg that has a ton of cytoplasm in it. And all that cytoplasm is going to be, right, the cytoplasm of that first cell of the offspring. And in that cytoplasm are all sorts of messenger RNAs and proteins that were derived from the mother. These are called cytoplasmic determinants. And these cytoplasmic determinants align within the zygote asymmetrically, such that when that zygote begins to divide, some of those cytoplasmic determinants go into one cell and others go into another. And that's sort of the beginning. Now, these cells are still very pluripotent. They can still go on to develop into lots and lots of different cell types, but we have at least the beginnings of asymmetry. Now, let's get back to here. So these cytoplasmic determinants are why the only way we can get the DNA from a differentiated cell like mammary cells, the only way to get it to direct the development of all other cell types was to put it into, take that nucleus, take that DNA and put it into an enucleated egg. Well, the reason we had to do that was because of these cytoplasmic determinants. They were necessary to reprogram that DNA and get it to divide properly and become all of the various cell types necessary to give rise to a new offspring. So the question is, what are these cytoplasmic determinants? What are these molecules that are in the cytoplasm of an egg that, number one, begin to asymmetrically divide, and number two, are necessary to reprogram the DNA here such that it can give rise to all of the various tissues or cell types of that organism? Well, we're right back here, induced pluripotent stem cells. Dr. Yamanaka, who discovered this, went through and looked at these cytoplasmic determinants and tried to figure out which ones were necessary for this reprogramming ability. And what he found were 4 genes that encode 4 important proteins. OCT4 is a protein that is a homeodomain containing transcription factor. SOX2 is an HMG domain transcription factor. MYC is a leucine zipper domain transcription factor, and KLF4 is a zinc finger domain transcription factor. So each one of these four, which is not surprising, all are transcription factors, specific transcription factors, proteins capable of binding specific sequences of DNA that turn on the activation of, turn on specific genes. What genes do they turn on? They turn on genes necessary to allow the proper development of this zygote into all of the various tissues and cell types of that organism. And so we're able to then take this, and instead of having to put a differentiated nucleus into an enucleated egg to reprogram it, we simply take the reprogramming factors out of the egg and put them into specialized cell types. The OCT4, the SOX2, the MYC, the KLF4. How do we do that? We use retroviruses that have been genetically altered to have the genes for each of these four proteins that then are used to infect these cells. And when these viruses infect the cells, they incorporate that DNA into the cells. Those cells now make these four proteins and make it there. Now, I should make sure that you understand that these cells already have the genes that encode each one of these proteins, but they're all turned off. Those genes are turned off, and so they're not being produced. When we do this with the viruses, we're turning all of those genes back on. We're getting production of those proteins, and thereby converting these specialized cells into pluripotent stem cells, very similar to embryonic stem cells. So the distribution of cytoplasmic determinants is asymmetrical in early development. The next way in which cells become different from one another during development is via inductive signals. So if we take a look at an early embryo that's made up of 32 cells, cells divide in very exact process such that every single 32 cell embryo will look like this. The cells don't just divide randomly. Which means the neighbors of these cells and the neighbors of these cells are different. Okay, the cells now are different based on who their neighbor is. Which means if these bottom cells here secrete a signal, like retinoic acid, for example, then the only cells that will receive that signal are the cells right next to it. So here we have a cell here that secretes a particular signal. That signal is received by its neighbor cell, and a signal transduction pathway occurs that leads now to this cell here producing activators that no other cells are producing. Right? These cells aren't producing it, these cells aren't producing it, these cells aren't producing it. Only these group of cells that have received that signal are producing a particular activator, and therefore going down a new developmental pathway. Retinoic acid is an inductive signal. Retinoic acid, when secreted by one cell type, is received by another cell type. And now retinoic acid is a hormone, a steroid hormone, which means that it can pass straight through the membrane. It doesn't need to bind to a receptor on the cell surface. Gets into the nucleus and binds to its receptor called RAR-RXR. And when it's bound to its receptor, it turns on a set of genes, okay, thereby taking the cell down a new developmental pathway. Now, retinoic acid, among other things, is involved and coordinates limb development. So here's retinoic acid. This is the mesoderm. This is one tissue type. And over top of that is another layer of cells called the ectoderm. And when retinoic acid is excreted from the flank mesoderm, there is a group of cells that receive that signal that then start producing another molecule called sonic hedgehog. Sonic hedgehog then becomes something called the zone of polarizing activity. And between continued retinoic acid signaling and this zone of polarizing activity, we get the differentiation of cells that will then go on to become limb. Now there's a really cool experiment to demonstrate this. What scientists can do is here, this is a chick embryo, and here is what's called the limb bud, that developing bud. Here's that zone, that sonic hedgehog that has been produced as a result of the retinoic acid signaling, and then you get the eventual production of a wing limb, so the normal limb of chicks. If you were to take a little teeny bead and soak that bead in retinoic acid and implant that bead in a different place on that limb bud, right? So here's where normally retinoic acid is expressed, and now we're forcing retinoic acid to leak into cells over here. What ends up happening is not only do you get a limb bud going out this way, you get a limb bud going out that way as well. So you get a mirror duplication of digits of that limb, because that retinoic acid is an inductive signal that leads to the production of a limb bud. There is all kinds of famous induction events during development. One of the most famous is something called the Spemann organizer. And if we look at an early embryo, just before gastrulation takes place, and so this sphere here is many, many, many cells, and we're looking at the Xenopus laevis, so a frog embryo. And right here at what's called the dorsal lip, there's a group of cells that send out inductive signals that are received by overlying cells. And what this inducer does is it induces these cells to take on a dorsal fate. That means the whole back of the organism. So, and then all of the cells on this side take on a ventral fate. Now, I have linked to the Canvas site a video that shows a very famous experiment where you can take some of this dorsal lip tissue, some of the Spemann organizer, and transplant it over onto the other side of the embryo, and that will lead to the production of conjoined twins that are connected by the belly. It's a really cool experiment, and I recommend you watching that video. So, inductive signals are another way that asymmetries start occurring, that different cell types within a multicellular organism start producing different activators and therefore start producing different sets of proteins. Now, if we take a look at enhancers and regulatory sequences and control elements, we see that they have different rules. There are some, for example, where in order to get the expression or the activation of gene expression here, each one of the enhancers must be bound by activators. If you have just one activator binding, it doesn't work. But there are others where the opposite is true, where you have a gene, and so this is called PITX1, and this is a gene in a particular type of fish. And here's our protein encoding region, and here are the 3 regulatory sequences. And here's the interesting thing, is that this regulatory sequence is identical in thymus fated cells, sensory organ fated cells, pelvic fin fated cells, and then other cell types. No matter what cell type we look at, these regulatory sequences are identical. Okay, well, this particular gene is turned on if any one of these control elements is bound by an activator. And this is interesting. This allows for some interesting ways in which things can evolve. So in cells that are fated to become thymus, these cells produce an activator called X. Well, activator X. I don't know exactly the name of the activator, and I don't know if it's completely known yet, but that activator binds to this particular control element and turns on that protein, and that protein then will help make thymus cells. Well, it turns out that PIT-X1 is also involved in the production of sensory organs. But to turn it on, there's another activator, activator Z, and it binds over here. And when it does so, it turns on the gene. Pelvic fins. Also, the cells that are going to be part of the pelvic fin also need the PIT-X1 protein. But the way these cells turn it on is they produce activator Y that binds to this particular control element to turn on the gene. And then all the other cell types that do not require PIT-X1, they don't make activator X, they don't make activator Z, and they don't make activator Y. Now, why is this particular system interesting? Well, it turns out that the way this has evolved, if a mutation occurs within this particular regulatory element, the pelvic fin regulatory element, activator Y can no longer bind it. Which means that only in cells that are fated to become pelvic fins will no longer make PIT-X1. PIT-X1 still can be made in thymus-fated cells. PIT-X1 can still be made in sensory organ-fated cells. So what's going to happen here is that this mutation that occurs in the regulatory sequence of the pelvic fin here, okay, no longer activator Y can no longer bind it, PIT-X1 gets turned off, and now we have a situation where we have fish that cannot make pelvic fins, but still have no problem with their thymus, no problem with their sensory organs. And it turns out that this is important to the evolution of these fish. Some fish of the species live in the ocean and they need the pelvic fin. Some fish live in lakes, in shallow lakes, and the pelvic fin is detrimental because they're quite often on the bottom, and the pelvic fins get all caught in the bottom of the lake. And so they have lost, through mutations here that have been selected for, they had lost the expression of PIT-X1 in cells fated to become the pelvic fin, and so they no longer make pelvic fins. It's pretty cool. Now, coordinate gene expression, the idea that multiple genes are turned on in the same cell type at the same time. In bacteria, that happens because you have multiple genes controlled by the exact same regulatory sequence, right? They are part of the same operon. That doesn't happen in eukaryotes. Instead, in eukaryotes, you get multiple genes that have similar regulatory sequences, like what we saw in muscle development. That's called coordinate gene expression. All right. So in our next video, we're going to take a look at epigenetics. The idea that gene expression can change not by changes in DNA sequence, so mutations in DNA sequence, but instead by modifying DNA methylation and modifying and remodeling chromatin. So we're going to take a closer look at that in the next video.